So we welcome you back for today's second session. The moderator of this session is Dr. Anvesha Bhattacharya, Ramanujan Fellow, AINN, and the rapporteur is Ms. Ashi, MSc second year student at AINN. Taking the session further, we would like to introduce Professor Sohail Pervez, Head of Department and Associate Professor, Department of Medical Elementology and Toxicology, Jamia Hamdard. He is an established name in toxicology. His studies have explored the Professor. Okay. My apologies, sir. He is an established name in toxicology. His studies have explored the potential of dopamine D2 receptor agonists, such as paramipexole and ropinirole, in reducing cell death and mitigating mitochondrial dysfunction following ischemic stroke and traumatic brain injury. Furthermore, his research has provided insight into the interplay between COVID-19 and ischemic stroke, highlighting the neurobiological implication of the pandemic. With recognitions like Alexander von Humboldt Fellowship for Experienced Researcher, Leibniz Institute for Neurobiology, Germany, UGC Faculty Research Award in Medical Sciences, and many more. He also has more than 250 publications and 8,850 citations showcasing his contributions to the science. I request Professor Pervez to please come forward to the stage and share his scientific insights. The topic of his talk is Beyond the Maze, Leveraging Invertebrate and Vertebrate Models to Probe the Neural Basis of Cognition.
So, very good afternoon, everybody. Thanks a lot for this nice introduction. It's always been a pleasure for me to come to Amity. I have been coming here often. I've been speaking here about different subjects, different core areas that I have been working on. And I would like to thank especially Malik sir and Dr. Rachna for arranging this workshop, which is the need of the hour. Neuroscience is a very interesting and dynamic core area of research of mine, apart from toxicology. And I see some familiar faces from my institute too, which is good. So, थोड़ा light. So I actually discussed with Dr. Rachna about the audience that I will be getting for today's workshop. And she said that mostly it will be students of PG and PhD and maybe some postdocs. So if I can just, by the raise of hands, is there any UG student here? Okay. I remember him from my last lecture. Is there any PG student? Okay, few hands. PhD scholars? Okay. And postdocs? Okay, we have one postdoc too. And any faculty members who are part of the workshop, if there are any? Okay. So we have a mixed bunch of all. So what today I'm trying to do is I'm focusing or centering my lecture around this maze. But before I do, I think now half of you who are from Jamia Hamdard or from Amity know about the history of Jamia Hamdard. But for those who are not aware about Jamia Hamdard, just one slide about the brief history that we have. This institute was created under the Institute of History of Medicine and Medical Research in the 1960s. And Pandit Jawaharlal Nehru actually laid the foundation stone, and it was inaugurated by Mrs. Indira Gandhi. And then uh, we had a few example, pharmacy, Unani medicine, nursing. So these were the integral part of Jamia Hamdard when it was affiliated to the University of Delhi. In 1989, through an act of parliament and due to the efforts of the ex Prime Minister of India, Sri Rajiv Gandhi, we actually got the deemed status. We have a medical college now, which is in inception since the past 13 years. And very recently, we were awarded the Center of Excellence in Unani Medicine by the Ministry of Ayush. Just a very brief history. So, just to introduce like what exactly we are going to talk about. I'm sorry, I forgot your name last time. Yes, Abhijay. Okay, now Abhijay, you are my volunteer for this part of the section. So if I ask you a memory puzzle, and if I ask you this question, what would be your answer for this? I would say something in between. Because if I ask you, you will say yes or no or maybe. Okay. Now, when we are talking about this paradigm, then we are okay. So you have to now appear for a test. And the test asks you, okay, you remember that? Okay, great. What was it? If you want to share with us. Hara bhara dosa. So you see, this hara bhara dosa was so tasty that he remembered even after so many hours. Such a difficult question. So now I'm going to ask you a very easy question. Are you ready for it? You want to ask him? Wow, that's some bonding. I'm impressed. Please don't focus on the date, focus on the year. Okay, so this is an issue. Why? Because we have associated certain things with our perception. For example, if I ask him, what did he get in his last birthday? What was the farewell about his school was? Who was his best friend in school? All these questions will elicit immediate responses, not only from him, but practically from everybody. That means that for memory, there is some kind of association. Okay, let's now come up with something that is very close to us. And I now need answers to it in chorus, as loud as possible. Okay. So let's start by going with the name of the movie. Okay. I don't hear it loud enough. Very nice. You have wasted so much of your parents' money in watching movies, and you are so good at remembering them. Okay. Now let's take a different turn. We will now again say very loudly in chorus. I think I do very loudly in chorus about. I just took what I was given. I didn't choose it. वो कुछ कह रहे हैं. Okay. So escape latency is actually the term we use for a water maze. So that means when you will plot a graph to show that which set of animals took what time, you will not write water maze. Yeah, you will not write platform mil gaya. You will write escape latency. It's exactly the way we have designed that we will take a particular parameter in a way so that we are able to convince the reviewers and the audience that whatever we did made some sense. So apart from water maze, you have other uh, things like the novel object recognition that I just told you what is all about. And then you also have this CFC, contextual fear conditioning. So what happens here is you can see that the rat is in a cage and you give impulses of foot shock. So it actually will get a shock which is not lethal, but that shock is to startle the animal in a way so that a freezing behavior occurs. So this can be done in many ways. You can add a buzzer to it, that you give a buzzer and then it will know that the shock is coming. So it is getting prepared for shock. First of all, it will not know something hit him. Then after a few travels, it will realize, okay, the buzzer is there, the sound is coming, shock is coming later. So then it is preparing itself for it. So this is how all other paradigms are also there, like social interaction tests, social recognition tests, and there are huge tests that are there. But again, the important thing is how well the tests fit within your hypothesis is the idea that you should gather on. Then with this, you can create a wonderful study. You can corroborate your biochemical and molecular findings with your behavior. And then you can convince yourself only that whatever objectives you are looking at is getting fulfilled. So you have also the mazes. You have an elevated plus maze, which actually looks at the anxiety. You have the reward related behavior paradigms where you know that the food is in the other corner, and then you have the place preference test and all. The next one is Drosophila, fruit fly. The advantage of doing Drosophila is that you can have a transgenic system of Drosophila very, very easily. You can have two different sets of male and female, cross them, and you will have transgenic, which is very difficult to do in rodents because as of now, according to IAEC in India, you need a separate animal house if you want to breed and do experiments in transgenic animals. So this is a huge problem and also the costs associated with it. So Drosophila, which we know as fruit fly, this is very peculiar. And I have again a small video for you.
Step
Here,
1.
you cannot
Hold the
just take
fly
out the
belly up
brain.
on its
You need
thorax.
to take out the entire
Step 2.
head of the Drosophila.
Stick your forceps underneath the proboscis and then pull off the fly head. Step 3. Pull off the left eye while holding the proboscis.
Okay. So this is how
Step
you
4.
actually remove the head.
Pull off the right eye
Now,
while holding
I
the
had
proboscis.
very difficulty in convincing one of my PhD scholars. She was always complaining, see my colleague
And the brain is
only
there
has to take out
photographed.
6 heads of one rat for one group. I have to take out 250
That
heads.
is easy.
It's a huge job. Taking out 250 heads of Drosophila for one group, for one experiment, is something very, very difficult.
Step
Okay. So now you also can have behavioral studies within Drosophila. It may sound difficult, but you have a jumping assay and you have a climbing assay. So what we usually do in our lab is we have two different lines of Drosophila. We cross them together. One is having the GAL4 and one is having the A beta 42. And the resultant fly that we get is having a red eye. So this is the indication or confirmation that we are using a transgenic Drosophila. So we have to check each fly here, and usually there is a 99% success, and that the flies that you are using are actually transgenic in nature. So this is actually your climbing assay, where you can see that this is my wild type and this is my AD, and see how nicely the wild type are climbing and the transgenic AD flies, which have no treatment, nothing, they are just transgenic, see how they are behaving. So again, if you see very closely, you can see them moving very fast, very clearly, and here. Similarly, if you have the jumping assay, this also can be done. 7 meters high platform. Here also, that the experiment will tell you how it's done. And also, if you can replicate the behavior and then convince your supervisor or yourself with results that even the behavior is corroborating with your experiments of molecular biology, that's something really rewarding. That's something that we should go for. So it's just the model you are doing, in which context you are doing. If you can make a sense out of it, that is huge. So you also have the larval crawling assay in flies. In my lab, we have not done it. We have done jumping and climbing assay, but this is how you actually even can look at it. The third model that is there is zebrafish. Unfortunately, we don't have this facility in our department. I am hoping that within the next 6 months, we will be getting it. This also is a very, very interesting particular alternate model, and the similarities with humans and the significance has given it a top-notch thing. One of the other reasons is the clarity with which you can get fluorescence imaging done in zebrafish is huge, is again enormous. So again, usually the behavior phenotypes can get to study variety of areas. For example, ataxia, bodily posture movements. Imagine using a rodent model for ataxia is so difficult. First of all, getting the ethical clearance will be difficult, and to induce ataxia in rodents, and just imagine having a behavior phenotypes within zebrafish that will allow you to do a high throughput screening with zebrafish. So just to give you an example, there are many common behavioral assays. All the mazes, as you know, the Y maze, T maze, even the zebrafish also has a T maze where you can do it. You can do a bite test. You can do a diving response test. All of these tests can be done with zebrafish. So this is again one of the dark and light behavioral alteration assay. You know how it is. Usually in our animal house, we have a 12 hours light and dark cycle for the rodents. Even usually the ethics says, and the animal house strictly follows them. So you cannot just do your experiments beyond 6 in the evening in rodents until and unless it's a time-bound study where you would like to see the response every 6 hours, or you are doing some kind of live imaging of that particular animal over the course of 24 hours. So again, these conditioning where you have a preferred side, a vehicle side, these all can be done pretty easily in zebrafish. I think, sir, you already have now a zebrafish facility here. So you will be able to see. And I was looking at one of the speakers, Dr. Sucheta, I think from IGIB. She is one of the speakers here. She will give you a very detailed analysis about. Okay. Okay, great. Yeah, she is one of the speakers I could see. So, yeah. So you have, again, you can see the latencies, how you do it. You also have the exploratory biting of the animals. You also have the rotating drum concealment assay where you look at the behavior of the fishes, like how they are hiding. So you have the T maze. See how nicely they have designed the T maze. You have the tap elicited startle reflex assay. You have seen the fishes, they move, they stop, and they startle and they move again. So these things can be done very easily. And just to show you an example, yes, this one I was looking for. This is a typical zebrafish behavior if you have three different groups. And if you would like to see what is the effect of stress on zebrafish. So if there is no movement and they are mostly lying at the bottom, that would reflect to have a high stress. When you have the normal behavior, usually it is flip-flopping and majorly in between. What actually low stress means is it is not normal, it is not at high stress, but there is something which is there that you cannot exactly explain in a fish. Why? Because the kind of things that we feel when India wins a cricket match against Pakistan is something you cannot ask the fish about. So it's again something that is corresponding to a low stress. Low stress is not normal. Low stress means something is going on that is actually taking it a little away from its normal state. Yes, sure. I'm just finishing. I would just like to tell them a little bit about, yeah, this is my last thing. So this is the C. elegans that we have in our facility. And I will just briefly show you two different assays. One is your locomotor assay. Is it moving? Where do I get it? So you see, this is actually a head thrash assay. The thrash is actually the movement of the worm that swings its head or tail to the same side, and it is counted as a thrash. So this is something which is kind of attributed to a locomotor assay of C. elegans. C. elegans is again an alternate model where you cannot see it with a naked eye. You need a microscope to do it. So within this, you have the head thrash assay, and also you have a body bend assay. You can see this is the natural movement of a C. elegans. And if the body is bending or the swinging of the mid body, this is thought to be a normal move. So with this body bend and with this, you can analyze it and then do it. So these were the different models. Did it get stuck? देखिए देखिए फंस गया है। See, again, when we are looking at locomotory behavior, so locomotory behavior would mean that the particular normal movement of a rat or any other thing. So once you have the C. elegans, the thrash would mean which side of the body the head and the tail are moving. If they are moving on the same side, that means the locomotion is fine. If not, then there is a problem. So with this, you can quantify also and look at different treatments. If you want to induce something that will not take care of the thrash, then that would mean that you have induced something. You can even have transgenic. Drosophila and C. elegans are the best alternate models because they are having so many subsets of transgenic animals and forms that you can do a variety of experiments. So this is, I think, all about, and yeah. So particularly for uh, the group of participants who have two X chromosomes, this is the take-home message. We may have 10% brain bigger than you, but you make it work more efficiently. And that is why, since ages, it has been said that you create the perfect balance between the home and the office and everywhere. So, usually, the paradigm of neuroscience, the behavior is not there anywhere. It is an exception that has to be translated, the power of which needs to be deciphered. You will have molecular biology, cell biology, biochemical things in every sphere, everywhere. But behavior is very much on its own, dedicated to neuroscience. So I would like to thank the different funding agencies that are there and the real people who are behind doing this whole thing. And Dr. Swati and Sabha, they are in the Drosophila group. Nisha is there in the C. elegans group. And Pinky and Neha are in cell culture as well as in rodents. And rest all the boys, they are working with the rodents. So these are the collaborators who have been helpful over the years. And thanks a lot for. I hope I'm within time. Okay. Oh, sorry.
Thank you so much, sir, for this wonderful talk. And now the session is open for taking up questions. Yeah.
Yes, please. Mhm. Mhm. A- Anything is applicable in rodent until and unless you can really translate it into a quantifiable measure. Stress can be in terms of heat also. Anything that produces any kind of response that is not normal is classified as stress. We say it as oxidative stress. So the reason why we do it is there. But again, depending on, like, many people are now even like trying to do exercise with rodents as a therapy for many things. It's very difficult to make a rodent follow a paradigm of exercise. And even sleep studies, very difficult to make a rodent awake for certain time. So stress, what we were seeing here was not that we are inducing it. If you have different groups, if you are inducing, let's say, a toxicant, then the measure, the way it is saying at the bottom, will reflect a stress due to the toxicity of that particular xenomine. If you would like to give, let's say, a therapeutic compound, a repurposed drug or the Unani formulation, anything, if it is really making the stress go away, then that is the mark for you that, okay, this is my kind of protection that I'm. So it all depends on which particular area you are working. So that is why I'm again repeating myself that only doing the behavioral paradigms will not help. To get the real collaboration, you go back to the basics that this particular behavior is elucidated by this particular area or this particular portion of the brain. Then you isolate that particular part. Usually, whenever we are taking all the other organs, we are taking it as a whole. But for brain, we never take it as a whole. Either you will take the cerebral cortex or the prefrontal cortex or the amygdala or the hippocampus. Even if you are, there is this electrophysiology in live animals. There also, you would like to stimulate the ventral tegmental area, VTA I was showing you, or nucleus accumbens, and then see a response.
More thing. So this is the stress thing. Okay. So in case of locomotion analysis, here also it is valid, right?
See, for example, gait, and GAIT, gait, not GATE. Gait is one of the major features of rodents when you are trying to study Huntington disease. 3-nitropropanic acid is the agent for inducing Huntington disease. And as soon as you induce it with 3-NP, you will see that the gait of the animal is completely changed. So at that particular time, these parameters will not help. Stride length, stride width, stride of an animal going. So there also it's pretty easy. What you do is you color the paw of the animal and you take a white paper and you let the animal walk. So the color, you know what is the distance between each stride will be your stride length and stride width. And by that, when you compare, you will see enormous difference between a control group and a 3-NP.
Thank you, Professor Sir, for a wonderful talk. As always, we love to listen to you. I have something like you have work across the species, right? So you have different models and working on like similar paradigms on them. So I wonder that if you have think of finding out the strikeout things between the similarities and dissimilarities across species when you are mapping the behavior, any sort of behaviors.
So again,
Thank you.
this is one of the questions that usually is posed to us by the IAEC committees. They will ask you that why are you doing the ischemic stroke model in Vista rats and not Sprague-Dawley? And if the student is there, then definitely his answer would be, "हमें sir ने बोला था." And then if the supervisor or I am there, then I will be defending, I have published 15 papers in this particular area in the last 15 years. So this is again very different. For example, we now know that for diabetes, Zucker rats are the best. But again, availability is a problem. So even if you have any set of animals that are the best, it all depends on the availability, whether it is there or not. At the same time, it is again important, behavior comes secondary. Whether that particular animal is representing or showing the symptoms of the particular disease you are looking at. So disease biology, chronobiology is completely different. Behavior biology is completely different. So it is an open question. Even the reviewers may ask that why you have done this. It has been shown that this particular animal is not showing this behavior properly.
Thank you.
Thank you, sir. Thanks once again.
Thank you, sir. It was a very wonderful talk. And now I would request Dr. Anvesha to kindly felicitate sir with his memento. So, as we reflect on the invaluable insights shared by you, sir, we would anticipate the next speaker of the day, that is Dr. Sunil Kumar Khatri. So, he's Director General, Amity Foundation for Science, Technology and Innovation, Dean, Research, Innovation and Extension Studies, Amity University, Uttar Pradesh. He is internationally known for being an academic leader with an expertise of more than 2 decades in a well established name in the field of artificial intelligence, software reliability and testing, and data analytics. His exceptional skills have granted him multiple achievements, namely Award for Excellence in Education Leadership, the Indus Foundation during Indo Global Education Summit and Expo, IT Innovation and Intelligence Award, Knowledge Resource Development and Welfare Group, an exceptional leadership and dedication in research, Society for Reliability Engineering, Quality and Operations Management, the list can go on and on. Sir, we deeply value your presence here and eagerly anticipate the enlightening exchange of the scientific ideas and knowledge that you will catalyze. The topic for his talk today is Amity University Research and Innovation Opportunities. So I would request you to take the stage now.
Good afternoon, everyone. Wonderful delegates coming here for this workshop, 5 days workshop. I believe you all are enjoying it, and I can see the students also of the institution. And this is going to be enriching 5 days for you, and definitely a lot of takeaways will be there from different sessions. My session will be generic in terms of opportunities and the initiatives being taken by the university and other university campuses also in area of research, innovation, and extension activities, and also that what all you can utilize whether you are a student of Amity or you are a participant. But if you would like to use any of these resources and facilities or opportunities at Amity, definitely we are always open to hear to you. Now, when we talk about this Amity the group, our Honorable Founder President Dr. Ashok Chauhanji, who came back after 3 decades being in Germany and with a vision that let us start something for the cause of society, and he want to contribute through education sector. And today, with almost 3 decades being in this industry, Amity have more than 300,000 alumni all across the globe from you passing out from university. We have 175,000 students with us. 11 universities are last being established in Amity, Bangalore this year, and next year also 2 are in pipeline. Globally also we have campuses more than 17 this is a snapshots of the various campuses of Amity, and if you can see the latest being in Bangalore University, which we had started last year. And these are some of the presence globally, including San Francisco and New York, in US, London, Amsterdam. Tashkent is also one of our university campus, which we had established in 2019, and I got the opportunity to be there for more than 3 years. We have been in Mauritius, we are in Singapore, we are in Dubai, we are in Saudi Arabia. Also, we are planning to go over Kenya. We are already there. South Africa, we are there. These are some of the pictures of our campuses globally. At Amity, we have 150 plus institutions with focusing on more than 300 programs ranging from every domain apart from core medical. When we talk about Amity research ecosystem, We have various departments to support in terms of research, planning from patents to commercialization to technology enabling center. We have IPR cell, Dr. Smita Sahu is sitting here. She is director of IPR cell. So this is also a part of our research ecosystem. We have DIT, Department of Technology Transfer, it has been sponsored by DST. We have a technology enabling center, again sponsored by DST. We have ACED, which is MIT Center for Entrepreneurship Development, where anyone can come out with their ideas and then can move on to the startups. There will be a couple of slides sharing details of how we can utilize these facilities, whether we are students of MIT or even participants from other who can utilize this incubator facilities at MIT for their ideas, ideation, and then converting into business plans. When we talk about the research activities of university, more than 30,000 publications are out of this, more than 450 research projects being running across the various campuses. We have more than 35 Center of Excellence at MIT. 3,500 plus case studies have been developed. Many have been bought by international universities, even from European countries. We have more than 2,100 patents filed and more than 335 being granted out of it as of date. We have more than 30 technologies which have been transferred and commercialized to the industry with the help of Department of Technology Transfer at MIT. We have Nobel laureates regularly visiting at MIT and interacting with their students. So Richard John Roberts Hebb Jr. and on his name and in his presence, the Center for Genetically Modified Organism has been established. Then Dr. Kajita has been here, and in his presence, Center for Particle Research Physics has been established. We have more than 50 high-end research labs at university. Participants and students, all of you can utilize and should utilize these facilities at MIT. We have MIT Center for Artificial Intelligence, which has been established last year, and with one of the largest capacity of processing power in India today. We are proud to mention it. As mentioned, we have a technology enabling center which has been supported and established with the help of DST. More than 32 scientists from MIT are in top 2% list of Stanford, more than 300 honorary adjuncts. Last figure which you see is the largest in India. Any university doesn't have more than that we have in terms of 40 Ramanujan and Ramalingam fellows. That is the highest with any university in India. As I talked to you about it as in previous slides, that at MIT we have a department for technology transfer and with the also a technology enabling center, which helps and facilitate in terms of your ideation, and also once your patents have been granted, how you can commercialize them, how you can reach to the market further. As mentioned, already more than 100 are in pipeline, and talking to the industries has been going on, discussions are going on for the commercializations of these technologies also. These are some of the technological advancements at MIT and breakthroughs that is happening throughout the years. Some of the very interesting research going on at MIT glimpse you can see over these slides, which is a very long list, and we can't go through them all. So these are just few of them at MIT, which are already commercialized. I'm only mentioning which have been commercialized, some of them. Now, across the university, as I had mentioned, that we have an ecosystem which includes not only foundations such as MIT Science, Technology and Innovation Foundation, MIT Foundation for Science, Technology, Innovation Alliances, IPRCL is there, ACED is there. But in other university campuses of MIT also a lot of research activities are going on. And if you see some of the examples in other universities, campuses, we will be sharing. These are some of the pictures of infrastructure. At MIT University Rajasthan also, we have got a very good project funding for PERS. We have a lot of equipments have been there working, and in fact, in Haryana also, we have a lot of facilities being developed through these funding, which have been granted from DST, DBT, to name some of them. Punjab is also has a very good infrastructure system that we have MIT University Mohali. we have built a satellite ground station at our Amity Dubai campus, which is a very beautiful campus, being mentioned by the in a magazine as one of the top 5 campuses of Middle East Asia. Some of the glimpses of infrastructure facilities and lab facilities at Amity, and the participants are invited that they can collaborate and use these facilities at Amity. As I was mentioning about that, we have a very great elaborative ecosystem, but not only for research facilities in terms of projects and publications or copyrights, patents, but also in terms of startups being motivated, as ACD is one of example, which handles, in support for your ideation. And then incubators, we have an innovation incubator, which helps you to take to the startup stages. We have more than 17 virtual incubation centers also through this AII. We have more than 200 business ventures and 40 million has been raised in investment. So any of you, if have any idea, definitely you want and to reach them, definitely we are always helpful and always be happy and available to help you with your ideas for startups, for commercialization. In fact, in the inaugural, Honorable Founder President was mentioning about IPR cell, and Dr. Smita Sahu is also there. So all the participants also, if you have any idea which you want to get patented and you want support system, we have this wonderful department, and she and her department has worked so hard. More than 2,100 patents being filed, more than 335 patents being already granted. So her team is always there to help you. If you have any ideas, you want to write a patent, how to write a patent, how to prepare a good document for it, and at a later stage, when it is in the stage of granting, how to commercialize it. We have DITT department. You want to get the help, you want to make a startup out of it, we have our innovation incubator. So a whole research ecosystem is there to help you if you want any of these opportunities to avail. I will move into the last aspect where we talk about the publication aspect also. If you see from national's perspective, in last 10 years, more than 18,57,000 publications have come out. And if you see some of the top places where publications are going on in India and the collaborators internationally with whom they are participating and publishing. And this is a slide for our Amity. If you see, we have 41% more citations of our researchers than the average of India. So our publications from faculties, from students coming from MIT are more sought after, more read, more cited. So it talks about the quality of publications of the work also going on it. And also, our Honorable Founder President always says that we as a university want to provide a research and innovation driven environment, all the support which we can do. Students, it's a wonderful opportunity for you to see and interact today and next 2 days also, when this workshop is going on with the speakers, get out some with ideas what you can bring into, and definitely the ecosystem at MIT University can provide you if you want to convert any of these into startup ideas. Thank you again for this patience hearing, and we look forward for any of your support that you look forward from us, any of the ideas from you for patents, for startups, for commercializations at a later stage, and even joint publications also with our students and our faculty. Thank you, everyone.
So much, sir. Questions are invited from the participants. Okay, we can go ahead.
So thank you so much, sir. The talk was really, really informative. And now I would request Dr. Anvesha Bhattacharya to felicitate sir with his memento. So last but not least, we have amongst us Dr. Smita Sahu, Professor and Director, Amity Intellectual Property Rights Cell, Amity University. And she's also a certified IP trainer from Japan Patent Office. Her research interests are intellectual property rights, biosafety, and bioethics. She did her PhD in life sciences from. Okay.
So I won't take more than 5 minutes of yours. I really respect your time. So I'm Dr. Smita Sahu, and I'm heading and serving in Amity IPR cell with the mission mandate of our honorable founder, President Sir. We are here to augment all IP filings with the portfolio of like trademarks, designs to protect the same and safeguard it. And we extend all the functional, structural facilities, support to our scientists and inventors towards filing it. And you'll glad to know that Amity University was the winner of National IP Awards 2020. And also our school has won the in Atoll Tinkering Labs. And also a lot of industrial IP awards have been facilitated and given to us. I'm here just to come up with an idea because this particular area holds a lot of IP potential. As you might be aware of, like the neurosciences with the advent of AI as well, you have a lot of prospect like the neurological, we have the neurobiological diagnostic methods and data processing of the neurological signals, or, you have computational models as well. So with the advent of AI, a lot of prospect is there. So we are always there to safeguard it. If you need any assistance, any facilitation towards it, because as our nation wants, to be among the top GII within the top 25 ranking, all the innovators' ideas needs to be protected and safeguarded. I will just quote it one word again. Once an idea is just come up to you, it's not that, you have to just come up with an experimental validation results and then to come and file it. There is a provision as a provisional filing. With a 10 or 20% of your idea, you still can protect it as a provisional specification. And over the period of time, 1 year will be given to you by which you can come up with your experimental details and file it as a complete, which gives you a grant status. I'll just brief you in this particular area in Amity University. We have filed a number of patent applications and many of them have been granted. Recently, few of them were like herbal nanoconjugates were given for the treatment of the Alzheimer's and Parkinson's. So likewise, a number of potential is there. So I welcome you all. If you want to be a part of such kind of research and endeavor, from the Amity University, we're always a call away for all of you. I'll share my details here only. If I can be of any help, I would be grateful. And all the best for your workshop. Thank you. Any questions? So I can just. And also the copyright can be filed, like we have come up with the neurological behavior, the perspective, the methods, the questionnaire, the scaling, the parameters, that all can be safeguarded in the copyright. So a number of ideas there, you always need to protect it, safeguard it, else someone else can misappropriate your name. Okay, thank you. I will drop my mail ID and my phone number. So just a call away for all of you. My mail ID is sahu@amity.edu. sahu@amity.edu. And my phone number is 9810 73 9810 73 2200. Thank you so much.
Thank you for illuminating us, ma'am. Now I'd request Dr. Anvesha to felicitate her. Ma'am.
So friends, you have seen how interesting that is from the presidents. We all miss such things when we are at many places, but sometimes it becomes facilitator. So with his experience, our respected founder president, he has created all these atmosphere. See, the Dean Research has come here to enlighten you the what we are here. Somebody head of the department of IPR. So the point is, if you have any such thing, it is not only you. Through you, we are trying to spread the message Not only to you, of course, our students are also here. Even they sometimes may not know. So if some of you are teacher or postdocs or somewhere, you can tell to the students, yes, many things are happening. And this is how any institution grows. Not only that, when you come up, you become a director, you become a faculty, you become somebody in the society. You have to know how to grow a society. That is very important. With this, I thank you. And then we spread for the lab continuation. Thank you, everybody.